Thanks everyone for coming out. Familiar faces, but for those of you who I have not seen before, the way we do this is we speak up here for about 30-ish minutes, and then I'll open it up for a question and answer, and then you'll get your chance to ask questions you may have. So tonight we have Dr. Adam Greer. He is an associate professor in the Department of Marine Sciences at University of Georgia, and his lab is based here at the Skidaway Institute. He earned a bachelor's degree from the Department of Biological Sciences at Vanderbilt University and a PhD in marine biology and fisheries from the University of Miami. He joined the Skidaway Institute in December of 2019. He is a biologist interested in the oceanographic, life history, and behavioral processes controlling the abundance and spatial distribution of zooplankton populations. Please welcome Dr. Greer.
Thank you.
So to start off, can you just give us an overview? What are zooplankton? What classifies as zooplankton?
So zooplankton are animals in the ocean that essentially have very poor swimming ability. They cannot swim against the currents, like the common oceanographic currents, and they kind of fall under this category. I guess I can start showing slides, right? So I have one slide that kind of introduces this concept. So plankton is a general term for any organism that can't swim against prevailing currents. And it comes from this Greek word planktos, which means wanderer or drifter. It's actually the same word that makes up the word planet, because planets like migrate across the sky and they don't form constellations or anything like that. So yeah, plankton, when they're first kind of described, it was like this phenomenon, like, wow, these animals are just out there drifting around. How do they survive? It's kind of still a mystery at some level. But yeah, there's kind of two components of the plankton. One is the phytoplankton, which are the organisms that are single cells or they're colonies, and these organisms perform photosynthesis, so they convert light energy into complex molecules and they produce oxygen. And then the animal plankton are the ones on the left side here. So this includes the group that is plankton all the time, so they don't have any sort of metamorphosis and they just drift around for their entire life cycle. This includes things like copepods, which are those little insect-like things on the far left, a bunch of different gelatinous organisms that are down at the bottom. And the other part of the zooplankton are the meroplankton, which are the larval animals. So basically every animal in the ocean spends its early life stages as eggs that are just floating around, and then they hatch into these larval organisms that are the early stages. A lot of them die off, but eventually some of them make it, and then they have a metamorphosis into something like a fish or a lobster or a crab. And so really, zooplankton are the thing that all these different ocean animals have in common, is they have this life stage that's just drifting around and sort of a mysterious part of their life cycle, because we really care about them when they're adults and we go fishing for them, but we don't know quite as much about them when they're young.
So can you speak a little bit more about the importance of zooplankton? Why we care about zooplankton, why we should care about zooplankton?
Yeah, so I mean, first of all, they're really pretty to look at. I mean, as you can just see here, but also they are kind of that link to the multi-billion-dollar fishing industry. Every fish that we eat in a restaurant or catch in the ocean at one point was a larval form, and during that time it was interacting with all these different zooplankton groups, and somehow it made it to a certain size where it basically escaped that lifestyle and started swimming around and living its adult life.
Yeah.
So I feel like we have to at least care a little bit about them. For a purely economic reason, if we care about fish, we have to care about zooplankton. They're part of that whole ecosystem.
So understanding which zooplankton are there and where they go has an enormous impact on what we can expect fisheries to look like in the future.
Yeah, absolutely. So, I mean, I didn't mention this, but those meroplankton stages, for a lot of ocean animals, it's the primary phase where they disperse to new habitats. So a lot of people are very interested in, what are the ocean conditions that allow, say, a population of coral reef fish to make it to a new environment, or even the corals themselves produce planktonic larvae that then are dispersed by the currents into another habitat. So this is called like population connectivity, and it's a pretty big topic in trying to understand how we conserve ocean habitats? How do you promote the movement of populations around? And usually it involves some component of the larval stage or the zooplankton.
So you mentioned that zooplankton are not very good swimmers. So how do they control where they go? Is this what determines that?
Yeah, that's a great question. So, I mean, it's not like there's just some animals that can't swim at all and then they all of a sudden can. It's like a continuum. And zooplankton are kind of at this level where they have a little bit of control, and so they can move up and down in the water column okay because the currents aren't quite as strong in that direction. And so there's a lot of studies where people have shown that crab larvae, for instance, the ones that are on the bottom, those are different stages of larval crabs. They can move in relation to the circulation around estuaries and actually stay inside the estuary or be dispersed offshore depending on where they move vertically. So these organisms, like, they've been around for millions and millions of years, so they're in tune with what are the oceanographic processes that can allow them to complete their life cycle or allow them to be like dispersed out into the middle of nowhere.
So I want to talk about your research. Just to start off broadly, what are some of the questions that you are interested in and your lab is interested in?
Yeah, so we study zooplankton ecology and biological oceanography, which basically means we want to understand how the ocean conditions, so the temperature, salinity, the structure of the water column influence the different organisms in the environment. And to do that, we do a lot of work with imaging systems. So all these pictures you see here are taken with this imaging system that I'll show you here. But so the thing on the left side is one of the systems that we use a lot in our lab. It's called the Modular Deep Focus Plankton Imager. It's kind of a mouthful, but the reason it's called deep focus is because the way it works is that you have a camera in one pod. So on the left side, that's one pod that has a camera, and the other side has a light. And so the diagram on the bottom left is kind of showing how that works. And so the light goes through a very small point source, and then it hits a plano-convex lens, which is flat on one side and round on the other. And that makes all the light beams go parallel to one another. And then it hits another lens and it gets focused into a camera. And so that technique allows anything to move around in that imaged water area, and it will be in focus. So that's a deep, it has a large depth of field, we like to say. And so that's allows us to look at a large volume, so everything's in focus, and we can just take a ton of these pictures, and we know exactly where each organism was. And with that, we can basically make a high resolution map of where the different organisms were. There's a lot of information just within the image about how organisms may be interacting and things like that. And so just as an example, this is a plot of some of the data that we would look at normally. So what I'm showing here is the distribution. So depth is on the y-axis, and then the x-axis is abundance. And so when those bars go to the right, it just means there are more organisms. And the black line is showing where a net system, what it measured as the abundance of that organism. And so that's what one of my graduate students, Patrick, is holding a net that we would use, and we tow that through the ocean, and it picks up a certain number. We know how much volume was sampled, and therefore we can calculate the abundance from that net. And so what you can see is that for something like this gelatinous animal, this is called a doliolid. I actually have an example of a few. If you want to actually look at them, they're easy to see with the naked eye, so you can just pass those around. They're the little barrel-like things. These are very important organisms for grazing on phytoplankton. They eat a lot of phytoplankton. But if you look at the graph, you can see that the line for what was estimated by the net is very low compared to those white bars, which basically means that the net is destroying a bunch of those doliolids. It's underestimating how many there are. With the images, we get a much more accurate estimate of these fragile animals like the doliolid. But something like a shrimp, it's more or less accurate. In fact, the net caught a little bit more than we saw. But with the imaging system, we can see little layers where the organisms tend to aggregate. And so that's really valuable information for understanding, how these shrimp may interact with different properties of the water column.
So you have the imaging device right now. is that a relatively new technology, or when did those come around? When did people stop using the net?
So people still use the net, and
Okay.
they will. In my opinion, they're always going to use the net because there's a ton of value in science for consistency, and so a lot of
Okay.
observations in various places all over the world have been made with nets, and so you don't really want to like replace a good thing. Like
Right.
they're still getting you good information, and there's value in consistency, so you can see signals of like climate or various long time scale patterns. But the I guess the technique has been around for quite some time. This is called shadow graph imagery, because you're basically making shadows by having a light and a camera. And I think it was invented in like the 1960s. And then only when the 90s came around, when people had, had digital cameras and just better equipment, then all these imaging systems started to kind of explode in numbers, and there's various types that all have their strengths and weaknesses. But ours is kind of like geared towards the larger zooplankton because of that large volume.
Are there any specific species? I know you've mentioned the doliolids. Are there any specific species that you're most interested in understanding and understanding their place in the food web and what they're eating and that sort of thing and where
Uh,
they're located?
yeah, so right now we have a bunch of projects, well, actually two projects looking at the role of doliolids specifically in the food web. And so this is a diagram I show in a lot of talks because it's basically showing kind of what I showed in the first slide, but in the food web form. So the base of the food web are all those phytoplankton at the bottom. And what we like to consider kind of the classic food web is what's on the left. So it goes from phytoplankton, it's eaten by copepods, and then those copepods are food for these forage fish like anchovies, sardines. These major fisheries all over the world are primarily eating zooplankton, the small fish, and then of course the small fish are eaten by the bigger fish. But what we're really interested in is trying to understand, when does the food web shift over to something like on the right, where the doliolids are the primary grazers of the phytoplankton. And we think that the doliolids, because they're fragile, they're gelatinous, they're low in carbon content, which is essentially meaning they're not quite as good a food for fish and things like that. Does it favor a more gelatinous dominated food web? And so right now we've collected a bunch of data in the South Atlantic Bight just off the coast of Georgia, but also in the northern Gulf of Mexico. And we're kind of comparing these two ecosystems to figure out, what are the conditions that lead to the gelatinous dominated food web versus kind of our classic food web. And we really care about the classic food web is what fuels all the fisheries all over the world. So that's kind of what we would like to see, I think, for the most part, is that. And we want to just understand, what are the natural processes that lead to that shifting, or can they coexist in different parts of the water column? So with our imaging, we should be able to detect things like that, like why are the doliolids dominant in certain parts of the water column versus others.
And the South Atlantic Bight typically is one of the areas that is less researched than other areas. Is this research and these sort of questions, are they being addressed by other research groups in the Northeast or on the West Coast and that sort of thing? Um, is the South Atlantic Bight and the Gulf of Mexico, that area, are they underserved in terms of these research studies?
Um, as far as the first part of your question, there's not, I can't really think of a research group out there that's doing the exact same thing with the
Sure.
type of this
Yeah.
like setup. But the way we kind of framed it to get the funding was that we have these wide continental shelf areas in both environments, but very different features of the ocean. So as you can probably imagine, the northern Gulf of Mexico gets a ton of river influence from the Mississippi River, a lot of other rivers in Alabama, whereas we get some, but it doesn't actually make it out to the shelf that far because the shelf is so wide and we just don't have as big river sources as the northern Gulf does. So that was kind of our way to say, like, look, it's the same latitude, very different oceanographically, so we can kind of do this like comparison and figure out what are the main causes of the food web shifts. And we can be pretty confident that they're actually connected by the Gulf Stream, and so they should be getting similar populations,
Yeah.
and they do have similar groups that are found in both environments.
Okay. So you have the toad system, which is Very fascinating, very interesting. And that is taking photos, and then you're receiving those photos. How do you go through those, and what are you doing with those photos once you get this ginormous? How many photos are you getting at a point? I mean, it's
Yeah.
many per second, right?
Yeah, it's about 5 per second, and so we tow the thing on station. We basically go to a station and tow it around, and usually that's about an hour. So we get like 10 to 15,000 images per station. At the end of a cruise, we'll have hundreds of thousands of images.
Yeah.
And so they go through. We have a lot of software that will go through and like uh, cut out. We call it like segmenting the image. So it takes a big image and it cuts out the little things. So I can just show you like an example of what a big image would look like. But I'm skipping ahead. But like something like this. So this is from the northern Gulf of Mexico. You can see all those doliolids are there in the image itself. There's a lot of just stuff in the water. This is actually something called optical turbulence. It happens when you go through a strong change in temperature or salinity. And so a good example is if you're at the airport and you see like the jet going off and you see how it looks kind of shimmery above the jet, it's because there's so much heat coming out of the engine.
Right.
It's the same phenomenon in the ocean, but it's based on salinity, so or temperature of the ocean. And so you have like a very cold, salty layer on top or below a very fresh and warm layer. At that interface, it'll be like really turbid and it'll look kind of like that because it's deflecting the water or the light is being like disturbed. And so, but this image is has already been processed a bit, but software would go through and actually cut out all those little doliolids. And this is a larval fish in the bottom right corner, and there's actually another one up there in the middle. Those are larval flounder or flatfish. I have one of those too. I can pass around. So just to queue up all those things I have. So this is a larval flounder from just off the coast of Georgia. Yeah, and so that one's I like that one a lot because it actually has pigment. You can see these little pigment spots on one side of the fish because it's very close to metamorphosis. And larval flounders are really interesting because they swim around as larvae like this, like they're flat like this, and at some point one side becomes pigmented and the eyeball will actually migrate through the skull to the other side and then it lives its adult life on the bottom like that. But the larvae are totally transparent, like paper thin, and they eventually, yeah, get to a certain size where they get some pigment and start metamorphosing. So it's an unbelievable metamorphosis that they go through.
Yeah, yeah.
But yeah, I think, did I answer the question?
Continuing with this, so once you have this photo, you break it down and you can kind of analyze which organisms
That's
are
right.
in this frame, right? So
Mhm.
here you've got doliolids and the flounder, but you also break it down into individual segments. What do you do with the photo like
So
this?
once you have, yeah, so the software will go through automatically cut out the things of interest above a certain, we give it a certain size threshold. We say like, we don't want to look at anything below 1,000 pixels. A pixel is just like a little square in the image. And then once we give it that information, it sizes all the stuff and it says, okay, everything above 1,000, I'm just going to box it and I'm going to put it in a folder. And then after that, we either look at it manually, which is obviously labor intensive, and we also are exploring a bunch of different techniques to do that automatically.
Okay.
And those have their own trade-offs where we're still kind of sorting out like what is the optimal level of detail that you can go into with these algorithms because we as scientists, we can look at these things and understand quite a bit about their taxonomy. There's limitations there
Right.
too, but we're generally much better at coming up with accurate identifications. But again, it's kind of there's trade-offs to each approach. Automated gives you more information faster, obviously, but there's a lot of steps to evaluate how good it is for different categories.
Right. Okay.
Because not every category is equally recognized by these algorithms.
So I know just from being in your lab and speaking with you that you have multiple, essentially versions of that pulled uh, system. So you've
Mhm.
got a benchtop imaging system, and then you've got a smaller one that's used for rivers, right? Or some.
So yeah, we had the main ones we use are the Toad system and the benchtop system.
Right.
And
Okay.
so the benchtop, I think I have a slide on that, but That's the one we kind of bring out for various demonstrations. But it is the same optics as the towed systems. There's a light on one side, a camera on the other, and we basically are capturing the organisms at sea and getting little video clips of them moving around with the idea that we maybe in the future can use these videos to develop training libraries for automated identification, because there's a real advantage to actually seeing. I mean, I learned stuff all the time. I looked at still pictures of a lot of these images when I was in grad school and stuff. But when we got this benchtop, I could actually see them moving around and like doing things, and it gave me a whole different perspective on like what they
Right.
could look like in the image, and like it's just a totally different view of how it operates. But yeah, I have little clips of stuff from the benchtop.
Here, I can pull that up. It might be
Oh,
behind there. Yeah.
okay, you have to go out.
Yeah. I also wanted to queue up these videos 'cause they're
Okay,
pretty
yeah.
cool.
I feel like we can go to them now. But yeah, so this is an example from the benchtop imager. And what you're seeing here is an amphipod. So this is a type of crustacean, and it's grabbing onto a siphonophore, which is a gelatinous organism. And these are very poor swimming animals, but they have really strong hooks, and they're often attached to gelatinous animals. Yeah. And then, yeah, so you can also see in the background, there's a lot of little copepods, the little things that kind of have the two antennae. So those are really, really abundant everywhere.
And then we have another one here too.
Oh, yeah, and this is a lobster phylosoma, which is a larval lobster, basically. And it's holding on to a hydromedusa. And I actually have one of those too, so we can look at those. But yeah, this is a lobster phylosoma. So you can see they're small, but they're not microscopic. You could definitely see them if you're looking for them, but the imaging system we use really makes them pop out so we can see them quite well. But when that's alive, it's almost completely transparent. It's very difficult to see, and yeah, it's yeah. So the lobsters actually, we have a lot of images where they're attached to bigger jellies than that. And for my postdoc, we wrote a paper about how you can actually see that they would they would be attached to jellies and like there'd be tentacles missing. And then I read some other study that had happened in like the 80s where someone actually they actually looked at it and they found that it would eat the tentacles first and then eat the main bell. So they're kind of along for the ride, but then also snacking on their host.
So I just wanted to ask like one more relatively broad question before I turn it over to question and answer. But um, there are so many questions left unanswered in this line of research right now. So with endless resources, time, that sort of thing, if you could answer any of these questions or see any of these questions be answered, what kind of questions would you be looking at? What do you want to be answered?
Yeah, I mean, a lot of the questions are really kind of like classic problems. And so what got me interested in this field, like I originally was really just interested in ocean fishes. And so I really liked fish. And then eventually I went to a research lab that was studying larval fish. And so I was like, okay, I'll study larval fish. That's fine. But now I'm kind of like more of I'm definitely like all in on the zooplankton now. But the reason I brought that up is because like the fish and fisheries are kind of the thing that drives a lot of research on marine life in general. And so there's this very old problem that's called, we talk about this a bit in the fisheries world, but it's called the recruitment problem. And it's this idea that if you go out and you survey the adult fishes, it's very it's very rare that the number of adult fish out one year predicts the number that come into the fishery the next year. And so that's what this graph is showing on the left side. It's the female spawning biomass. There's two y-axes, and then there's another axis that's the age to recruits. So those are the young fish that are coming into the fishery. And you see when you look at the spawning biomass, it does this kind of like nice, normal, it went up and then it went down. But if you look at the recruitment, so that's the number of the little fish coming into the fishery, these age two, it doesn't really match up at all. There's almost no relationship. And it just means that the environment is doing something that because we presume that all the fish are spawning. They're releasing eggs into the water column, but then something is happening that we have no idea about during that process that's leading them to be very few or a lot to show up from one year to the next. And so this kind of this graph on the right side is showing what we think is happening in those early stages. And so there's the y-axis is abundance, and the x-axis is time in days. And this is going from eggs to yolk sac to larvae to juveniles. And it's kind of hard to see, but each one of these little lines is showing the processes that we know are happening and how they're influencing those different life stages as they grow up. And you can see the numbers are declining greatly. So just a fraction of a difference in the survival rate can be the difference between a ton of juveniles showing up or not many. And so really that's kind of a broad motivation behind my research, is to say, like, okay, are there processes in the ocean that we can better understand to at least have some clue about what might be happening to cause higher or lower survival for those larvae each year? And we think, that the gelatinous population could be really important, those jellies, because they're so undersampled. If there's a lot of them one year, are they eating a lot of the larvae? Like, we just have no way to measure that. And so I think the future is really about getting better observations in that environment so that we can figure out what are the main factors that actually contribute to that variability. So we're not just like throwing up our hands like, oh, well, it's an impossible problem to solve, because it has to be solvable, but we just need better tools to observe what those little larvae are actually experiencing.
Sure, yeah. Well, are there any audience questions?
The water seems so pristine in your slides. Is it really that pollution free?
No, I mean, there's definitely pollution. We wouldn't really be able to detect most of the pollutants in the type of imaging we do, because you have to remember that each pixel in there is about 50 microns, so it's tiny, but most pollutants are almost invisible. It's chemicals that have been put into the water that don't show up as objects that would block the light between two pods. You know what I mean? So it's just the imaging is not the best tool to use to detect pollutants. But there's definitely pollution out there. I mean, Gulf of Mexico, tons of oil drilling. There's actually natural seeps all over the Gulf. It's kind of an interesting phenomenon. But yeah, there's hydrocarbons just seeping up out of the benthos all the time, out of the bottom of the ocean. So, but there also are microbes that eat that oil and those hydrocarbons. So the pollutants we tend to worry about are the ones that are forever chemicals, like mercury and things that just stay in the system and have no way of being removed.
You turn your mic off first.
Oh, turn it off.
Okay. Yeah.
It would seem like if you can, in addition to imaging, also measure physical and chemical characteristics and determine that there are differences that might help you predict how well the various zoal creatures will do.
Yes.
That you could then, over time, also use this to look at factors like climate change in terms of drilling down and being very specific about what happens in the microclimate that you're studying, but also maybe generalizing that to what's going on in a wider community.
Yeah, no, that's a great point. Yeah, the downside of a lot of new technology in general is that you kind of have to just see what you get at first, and then over time you can get to a long enough sampling period where you can actually see some kind of signal from longer time, like climate change or longer. But I didn't actually I can go back to it. I didn't really explain how the system works, but it's imaging with a bunch of other sensors. Talk about the temperature, salinity, dissolved oxygen. We added a PAR sensor, so that's detecting the downwelling light, chlorophyll A. And so we have this kind of suite of variables that we try to see what is the best predictor of the different groups that we're seeing. But yeah, adding more sensors is always something we want to do and things like that. So yeah.
So did you say that jellies are carbon poor? They have poor source of nutrition? What are they? What are they made of?
So what I meant by that was I should probably use the wrong word, but they're more like they're higher water content. So the carbon density or the energy density is low. And so sometimes the analogy I use is like if you're a fish and you have a choice between a cobepod or a small jelly. The cobepod is like a cheeseburger, and the jelly is like an iceberg lettuce. It's just not going to give you that much fuel.
Yeah, is there a difference between freshwater plankton and saltwater plankton?
Yeah, pretty big difference. So the groups that dominate freshwater look pretty different. There are some freshwater copepods, for example, but they are relatively uncommon. And so there's species that called water fleas or Daphnia. It's another group that's not that common in the ocean, but in freshwater, it's really common. So yeah, they look really different. The ocean is much more diverse in terms of and the ocean is also much more gelatinous. So freshwater, you don't see that many gelatinous organisms or jellyfish. For whatever reason, they don't tend to do that well. And there are some examples, like in biology, there's almost an exception to every rule. But yeah, the jellies don't see them in rivers. You don't really see them in lakes. But the ocean is super gelatinous.
As temperatures increase in the water, do these organisms have an opportunity, or can they moderate themselves and put them into a more friendly or temperature friendly situation?
Yeah, that's a great question. I can't say we know the answer to that. We do sometimes tow our system in day and night to compare the distribution of the animals between the day and night to see how much they would move based on the signal from the sun. But what you're talking about is kind of almost like a longer time process. And so I would say it depends on their food resources, their predators. There's a lot of factors that go into the way animals behave. And usually it's some sort of like happy medium between finding access to food and then avoiding predators, especially in the zooplankton, because you're out there in the middle of the ocean. You have no place to hide. Your only defense really is to like move out of the high light environment to get away from a visual predator, or short bursts of swimming away. But usually that doesn't work out too well.
We hear a lot about microplastics. How does that affect these creatures?
That's another great question. I have a collaborator, Jay Brandis, maybe you've heard him talk about microplastics, but that he's taking he's a co-PI on this project, so he's also out there, or not on the ship necessarily, but he has people out there who are collecting some of this information about like how many sizes and types of microplastics that are out there. But that's kind of it's a really a lot of people are interested in determining how many plastics get into the food web through the different zooplankton, because presumably they're eating all sorts of phytoplankton. They're probably incidentally ingesting some of them. So yeah, I wish I had a better answer, but I can tell you that it's a lot of people are interested in figuring that out, as it is kind of pathway into the rest of the food web.
Do you find, I'm thinking about like the spider crab and the cannonball jelly association, like missiles and everything. Do you find those kind of relationships? Or is it just all predation, like at that age?
Yeah, we actually published a paper like 5 years ago. It was kind of like, it was something that I wanted to do for a long time, but it was basically like, I took a bunch of these like images that I just had collected in a folder that showed all these interactions, and basically wrote a little paper about how with images you can see so many more interactions than all these things would be unknown to us if you were towing a net, like you just get a glob of, you know, the jellies and some crab, and you're just like, okay, I guess they were around, but you had no idea that they were actually physically like interacting. And so there's all sorts of examples of parasites, you know, the example of the jelly that's, you know, being eaten by the lobster larva, lots of examples where there's clear associations. There's a lot of fishes, so larval fishes that actually they appear to be hanging out near larger jellies as almost like a floating shelter. And so, I mean, there's a kind of well-known thing in fisheries that like, there's like fish aggregation devices. Like if you go on an open ocean, you just stick an object out there, fish will just show up over time. And so there might be something similar happening in the larval stage where they just kind of like find something a little bit bigger and it's some structure. So it's like a type of defense where if like a bigger fish shows up, at least they could like move around the object and hide, as opposed to being in the open ocean where a little fish against a bigger fish has no chance. They just can't outswim it. But yeah, it's kind of I think it's like a really interesting topic for like AI too, is like, could we feed AI a bunch of images and then have it like understand? That's kind of like what's happening now in the AI world, according to one of my colleagues. They're trying to design like agents or things that will like infer interactions over huge data sets without us. Like look at an image and infer some like purpose or some like systematic relationships as opposed to just how many are there.
Right.
Something similar to like what we do when we look at an image. Oh wow, that fish is oriented like this, and there's another jellyfish right here. What could be happening? Like that would be, I think, really cool if we could do that at a big scale, and then we'd learn a lot more about all the different interactions out there. At least get an idea from the AI what ones would make sense or which ones are most common, and then we could like take it from there.
Spend a lot of time with the benchtop imager.
Yeah. Well, hopefully it would come from the toad system.
Okay.
But yeah, the toad, like the toad system would get like millions of images, and then the AI could just say like, okay, what is there a systematic relationship between this organism and that one, and like how often? It's I haven't thought about it too much, but apparently that's what like modern day AI is able to like look at a picture and determine like that thing's in the background or like, you know, this chair. What it can actually understand the scene better than even like a year or two ago. Right. So it's changing like all the time.
Any additional questions?
What's the, you were talking about the need to understand the survival and predation and recruitment. What's the constraint to getting to the understanding? Is it simply time? Is it number of scientists involved? Do you need folks?
I would say it's a combination of factors, right? In the world of fisheries, the way it operates is a lot. They're dealing. It's a very difficult problem. We're actually talking about this in marine policy on Friday, but just understanding what's happening in fishery is very difficult because what you do is you go out there and catch a bunch of fish, you look at the sizes, and you try to determine like how fast is the population growing and things like that. And so that takes a lot of effort to just do that. And then a lot of the fisheries models, people that are working with those data, they rely on that data from the adult population. But it's a little bit outside of how fisheries are managed to talk about larvae, because a lot of them, I mean, to be honest, most of them die. You know, they're not, it makes sense if you're going to choose to allocate your resources, you focus on the adults because they're the ones getting fish. And so I guess I'm kind of long-winded way of saying that, you know, to discover some indicator of the fishery from the larval stage it takes a lot of just effort. And then also you have to get really good information about the fishery. So it requires a collaboration between fields that are
traditionally a bit separate, like biological oceanography and fisheries science are not necessarily doing the same thing. So I think the key really is for, you know, biological oceanography and fisheries scientists to like try to work together more and get a better sense of what each group needs. And so right now, the way fisheries operates is mostly based on the adults. And so it kind of is the future. A lot of people are talking about ecosystem-based fisheries management. So people are really thinking about this in the future, like how can we use other properties of the ecosystem to help us better understand the health of a fishery. So maybe we don't have to go out and catch a bunch of them, or we can at least like get some sense of it or something that's a little easier to measure potentially. But yeah, so I'd say it's an observations issue and then also a disparate fields issue.
Would you ever have enough young scientists coming up in those areas to keep looking?
I mean, I think that the technology is going to be a key part of it. That's sort of where, you know, that's what I'm really excited about is the technology can really make observations that were previously just not possible to do. So, I mean, a lot of these fields, they form based on whatever was the way that people observed at the time. And so it's not, most fields don't change overnight. So it's really about, yeah, I think young scientists are learning more about the technology, and there's going to be, you know, smart people who think about how we can use the data or the information from these technologies to maybe inform some of these decisions we make about the $5 billion fisheries industry. So, yeah, it's a great question though.
Anything else? All right, well, thank you again to Dr.